Welcome to today's session of ACCESS Presents. I'm Philip Lang. I'm an investigator at BC Children's Hospital and the co-lead of the cancer biology theme in ACCESS. And it's my pleasure to host today's session. So first of all, I would like to acknowledge that I'm speaking to you from the unceded territories of the Musqueam, Squamish, and Tsleil-Waututh nations. And as ACCESS as a whole, we respectfully recognize and acknowledge the relationship the First Nations, Inuit, and Métis across Canada have to all the land and to the Canadians live on and cherish. As we work to create, implement, and continue this pan-Canadian network, we would like to consider each of the 634 different First Nations communities, 53 Inuit communities, and 8 Métis settlements who have a unique historical, cultural, spiritual, and environmentally sustainable connection to the land that their people and ancestors have inhabited since time immemorial. And in ACCESS, we're committed to working and building relationships that include First Nations, Inuit, and Métis people. And this last part, to me, is really the key here. We can acknowledge all we want and keep acknowledging, but moving forward, in particular, in a network like ACCESS, it is really about action and ensuring that we shape the future together and that we ensure that all children have appropriate access and access that's culturally safe and appropriate to the care they seek. And so in that spirit, I would like to give the people on here who might not be fully familiar with what ACCESS is and what the spirit of ACCESS is, a brief idea. So ACCESS is a pan-Canadian pediatric cancer network that sets its goal to advance the childhood cancer experience, science, and survivorship. We're a network of researchers, healthcare providers, partners, and people with lived experience. And as this network, we stand united in a shared commitment to equitable access to research, to innovation, to care for children with cancer and their families in Canada. And one of the ways we support these objectives is by hosting free educational sessions like today's seminar. And with this, I would really like to welcome you to today's session. Maybe before I welcome you to today's session, as a general one, we'll have question and answers at the end. So please hold them, and at the end you can type them into the question and answer chat, and then we will be happy to address them. We have a number of experts on the panel today that can answer those questions, and I'll introduce them towards the end. So today we're gathered to learn about Prospect, Proteomics for Personalized Evaluation of Childhood Tumors. And this is really an initiative that has emerged, that it's been going on for several years at BC Children's, but then really flourished within the cancer biology theme of ACCESS, where it draws on a number of resources and really brings together activities across the nation. So it draws on the preclinical modeling program and also on the National Molecular Pathology platform, within which we have strategies focused on getting access to new diagnostic testing in the National Molecular Pathology Board, advancing the ability of less invasive liquid biopsies. We won't be speaking about those today. And then what we'll learn today about how we can complement personalized diagnostics, that is genome sequencing driven to date, by looking at the proteome. And with this, I would like to introduce our speaker, Dr. Kirsty Ferguson, today. She's joining us from BC Children's Hospital, where she is a senior scientist, a research associate, and leading the effort in really converting protein level information into the identification of treatment opportunities for individual children. And that's what she'll talk to you about today. Over to you, Kirsty.
Hi, everyone. My name is Kirsty Ferguson. Thank you for the introduction, Philip. And we're very grateful to ACCESS for the opportunity to speak with you today. And thank you very much for coming along to hear more about our work. So today I'm going to be speaking to you on behalf of the Lang Lab about how we're using a technique called proteomics to bring more information to guide treatments for children with difficult to treat cancers in Canada. And as Philip said at the end of the presentation, the whole Prospect team will be here to answer any questions you have. So, did you know that 20% of children with recurrent or hard-to-treat cancers still lack effective treatments? Cancer treatments for children have traditionally lagged behind adult conditions due to lack of funding and disease rarity. However, chemotherapy and radiotherapy leave children with long-lasting side effects, and therefore targeted and more kinder treatments are urgently needed for these young people. Fortunately, the development of next-generation sequencing is enabling doctors and scientists to find personalized treatments for children. So this means we can read all of the genetic code, or the DNA, in a child's tumor in a technique we call genomics. And you can think of this genetic code as a cookbook containing all of the recipes or instructions for our body to work. And using sequencing, we can detect if certain genes have mutations or duplications or deletions, which are mistakes in the recipes, which might make a patient's tumor more sensitive to certain drugs. And before these recipes then go to the kitchen, they have to be photocopied, or what we call transcribed, into a molecule called RNA. And we can also directly read this RNA in what we call transcriptomics by measuring if there's more or less copies of a certain recipe. We can this also helps us predict if there might be sensitivity to particular drugs in a child's tumor. And researchers from around the world are using these techniques, genomics and transcriptomics, to read the DNA and the RNA of children's tumors to offer medicines tailored to each child's cancer and transforming the care for children with cancer. Here in Canada, PROFILE, which stands for Precision Oncology for Young People, which is a driver project of ACCESS, is providing the hope of precision medicine. So from across Canada, children with difficult to treat cancers can be enrolled to receive molecular profiling of their tumor, so analysis of DNA variants and RNA levels. And the results of PROFILE have directly impacted patient management decisions for almost 1 in 4 patients. However, we believe there's more information. So after these recipes are photocopied, so the DNA into RNA, they are then sent to the kitchen to make the final product, which is called a protein. And for several reasons, we think that studying these proteins could provide complementary information to help even more children. So firstly, children's cancers have 14 times fewer mutations, so fewer mistakes in the cookbook than adult cancers. And this means that sequencing of the DNA may not always reveal clear treatment options. Secondly, as you've just heard, genes are photocopied to RNA, which are then the instructions to make proteins. However, levels of RNA and protein do not always correlate well. And there's many regulation steps that impact the final levels of proteins and how they function. For example, differences in how stable proteins are, or modifications, which can be thought of as like adding chocolate chips after the recipe is completed to the final product. And lastly, most drugs target proteins themselves, as proteins are the functional molecules of our cells. So the cells in our bodies make many different proteins, here represented by different colors. And these are made in different amounts, shown by the different sizes of the circles. And each protein has a unique function in the cell, all working in combination to do all the tasks a cell needs to survive and reproduce. And unlike genes, proteins are dynamic, so their levels change over time depending on their environment. And different cells make different combinations of proteins to perform their unique functions. So while neurons in the brain, cardiomyocytes in the heart, and hepatocytes in the liver all have the same DNA, they contain many different proteins in different amounts to perform their unique functions. However, changes to these proteins can turn healthy cells into cancer cells. And we believe if we can find the difference in the proteins between the normal cells of a patient and the cancer cells of a patient, we might be able to find additional treatment options. So using proteomics, which is the study of proteins, we can take a patient's healthy cells and cancer cells, extract all of the proteins, and then create maps, which we then use to spot the differences between the healthy tissue and the cancer tissue. And this helps us to understand what might be causing the cancer. So for example, in this example here, we can see that there's many differences, but one of these is there is more of this protein in purple in the cancer cells compared to the healthy cells of the patient. And we may know that this protein helps cells to divide and reproduce, for example. And we therefore then want to know, is there a drug that targets this protein? As this might be a way of killing the cancer cells without affecting the normal healthy tissue. And over several years, scientists at BC Children's Hospital have built up the expertise and techniques to make this analysis possible through PROSPECT, which is proteomics for personalized evaluation of childhood tumours. And the PROSPECT team is made up of scientists with diverse backgrounds, from bioinformatics to pathology and cancer biology. And within ACCESS, we're now establishing a workflow to bring proteomics to childhood cancer patients across Canada, with teams in Toronto, Quebec City, and Montreal. So first, we identify profile cases who may benefit from proteomics analysis. For example, where analysis of their DNA and RNA didn't reveal any therapeutic options. We then receive the tumor biopsy and dissect the tumor and the normal tissue. We then extract the peptides and run this on an instrument called a mass spectrometer. And then we perform data analysis to spot the differences between the healthy and the cancer tissue, and look for linkages between the proteins and linkages between proteins and drugs. And then lastly, we report this data to the molecular tumor board at the relevant hospital for discussion. And I'll now go through this process and the problem solving and progress which has led us up to this point. So firstly, when a biopsy is taken from a patient, so a small piece of tumor tissue during surgery, this can be flash frozen in liquid nitrogen, which is minus 196 degrees Celsius, which preserves the sample very quickly. However, for many reasons, it's not always possible to receive fresh frozen tumor tissue. And the solution for this is for us to be able to perform protein analysis on routine pathology samples. So a piece of tumor tissue is always fixed in formalin, and it's then embedded in paraffin wax by pathologists. And this can then be sliced very thinly and mounted onto glass slides and stained, which is used by pathologists to diagnose tumors, and it can be kept long term. So Verena and Georgina in the lab developed a protocol to extract proteins from these formalin-fixed paraffin-embedded tissue blocks with the same quality as proteins from cells or fresh frozen tissue samples. And this allows us to perform our analysis on routine samples and integrate into the clinical workflow. So next, we need to find ways of extracting the proteins from the tumor and the adjacent normal healthy tissue separately. And in some cases, there's a distinct boundary between the tumor and the normal. And this allows us to perform macrodissection, which is essentially where the tissue is directly scraped off the slide into a collection tube. However, some tumors are diffuse or spread out, or they're heterogeneous, which means that the tumor may differ depending on the different regions which are analyzed. And in this case, we need to find ways of extracting different areas of the tumor from the same sample. So to do this, Verena in the lab, together with pathologist John Bush at BC Children's Hospital, have collaborated to establish a microdissection method. So first, John marks areas of the tumor and normal tissue for dissection, and then Verena can precisely cut out these areas using a laser capture microdissection microscope. So here you can see an example of a stained tumor slice on a slide, which is 10 microns or 1/1000th of a centimeter. And this is the microscope that Verena works on. And you can see here a video of a laser cutting out a piece of the tumor tissue. And this then falls directly into a 96-volt plate below it. And this allows Verena to collect many samples that can all then be processed at the same time. And optimizing this technique in the lab means that we can adapt the dissection method depending on the sample that we receive. So next, the samples are processed and analyzed on an instrument called a mass spectrometer by our mass spec magician, Jehan. And in the Lang lab, we are very lucky to have state-of-the-art instruments, including a new mass spectrometer called, which we call the Zoom, which was the first of its kind in Canada and funded by generous donations from the BC Children's Hospital Foundation. So after pieces of the tumor have been cut out and collected, the proteins are digested using an enzyme, which cuts them up into little pieces called peptides. And to explain this technique, which it has the full name liquid chromatography mass spectrometry, Jehan uses the analogy of a coin sorting machine. So we inject the peptides, which can be thought of as coins of various sizes and values. And these are then separated depending on their size and value. And the machine then counts or quantifies how many there are of each. And then by comparing this data to databases of proteins, the peptides are put back together to determine how much of each protein is present in the sample, something we called protein inference. So now we have this data. We want to spot the differences in the proteins between the healthy tissue and the cancer tissue of the patient. And then the data goes to our bioinformaticians, Augustina and Ennis. And they have developed an analysis pipeline which allows them to quickly identify proteins that are at significantly different levels between the normal and the tumor tissue. So, for example, this is called a volcano plot, where the dots in blue are proteins with a lower abundance in the tumor compared to the normal, and the dots in red are proteins with a higher abundance in the tumor than the normal. And these differences are what we call protein targets. So now we've identified our protein targets, but in order to identify truly personalized treatments, we need to compare the results to other patients with the same tumor type. And to do this, John is developing tumor tissue microarrays, or what we call TMAs, for different cancer types. And Verena can then dissect each of these samples using the laser capture microscope. So you can see here a slide similar to the one before, but instead of one tissue sample, this has many tissue samples from different patients. So each of these circles, which we call a core, is a sample from a different patient with the same cancer type. And this then enables us to see what is the abundance of the protein target in the patient we're analyzing compared to a cohort. So for example, for this protein, we can see that there is a higher abundance in the tumor compared to the normal. And when this is compared to the patient cohort, it is in the 95th percentile, which means that the abundance is higher than 95% of patients in that cohort. And this gives us further confidence in this protein as a potential therapeutic target. Next, we want to know if there is a drug that targets our protein of interest. And the problem here is that not all of these significantly different proteins are associated with approved drugs. So to solve this, Augustina developed an analysis pipeline to match the identified protein targets with drugs which are commonly used or requested in Canada for children. And this allows us to quickly and routinely filter our data for targets with associated drugs that we call actionable protein targets. Lastly, I perform a literature review on all of the actionable protein targets, and we convert this data into a condensed report of the key therapeutic targets. And we then present this to the molecular tumor board meetings, where we discuss the results with the clinicians and genomics analysts. And importantly, this whole process has a rough timeline of 6 weeks, which matches the timeline for reporting of genomic analysis of tumours, which helps us integrate these 2 bits of complementary information for the patient. So last year, this whole workflow was completed, and it was shown to identify a new drug target for a patient which was invisible at the DNA and the RNA level. And this study was done by a multidisciplinary team across Canada, led by postdoc Georgina and PhD student Tarek, and it was published last year. And the patient had a very rare cancer called settle, or spindle epithelial tumor with thymus-like differentiation. And they received a treatment based on genomics analysis of their tumor. However, unfortunately, the tumor developed resistance to this and they relapsed for a third time. And at this point, the team then performed proteomics analysis to see if they could find additional treatment options. So macro dissection was performed on the formalin-fixed paraffin-embedded tissue. And then the analysis revealed increased levels in the tumor of several proteins involved in how the cells make energy, including an enzyme called SHMT2. And interestingly, the team found that SHMT2 could be inhibited by a drug called sertraline, which is an antidepressant with a known safety profile in children. For this case, fresh frozen tissue was available, which meant that we were able to test how well the patient's tumor responds to this drug sertraline in the lab, together with the BRAVE initiative at BC Children's and the ACCESS preclinical modelling programme. For example, James Lim's lab have established a way of using chicken eggs to grow small pieces of the patient's tumor in the lab and then test the drugs found from the analysis. And they found that 5 days of sertraline treatment significantly reduced the tumor growth, fitting that this may be a potential treatment option. The patient's tumor cells were also expanded in mice by Donna Senger's lab, and the expanded cells were used to test response of the tumor in zebrafish larvae by Jason Berman's group. And this also confirmed that the tumor growth was significantly slowed by the sertraline treatment. So together with the proteomic analysis, these data led to the molecular tumor board, suggesting a 3-month innovative therapy trial with sertraline, which the family decided to pursue. And CT scans were taken before, during, or after therapy, and this showed there were notable decreases in the monthly tumor growth rates at the end of the treatment, with no new parenchymal lesions forming. And this showed for the first time that proteomics could provide actionable information beyond the genome within a meaningful time frame to directly help patients in the clinic. So far we have analysed 5 patient tumours from across Canada and reported our findings to the molecular tumour boards. And this includes a case at Sick Kids that was analysed in collaboration with Mike Moran's group at the Spark Biocentre. And through ACCESS, we're implementing similar programs at hospitals across the country. And we hope this will help more children find access to innovative treatments. And part of making proteome analysis available to all Canadian children with cancer is making sure that we have robust ways of interpreting the data that is processed at different hospitals in different locations, which will have slightly different methods and different equipment. And to do this, the ACCESS Proteomics group, which is part of the National Molecular Pathology Platform, is working to analyse and compare samples from hospitals in Vancouver, Toronto, Quebec City, and Montreal. Another vital aspect of moving this research towards patients is knowledge translation and dissemination or sharing. So to make sure that the creation of this new knowledge has a tangible impact on patients' lives, it's so important that we find ways of sharing research outside of the scientific community to reach policymakers and those living and affected with childhood cancer. So academics often think of knowledge sharing as traditional routes of presentations, papers, and posters. However, there's more creative methods which are needed to make research more accessible. And in the Lang Lab, we're using various ways to share our research to reach a wider audience. So one example is Georgina and Tarek, who co-led the study I mentioned on analysing the patient with the rare settled tumour, took part in a podcast series run by BC Children's Hospital Research Institute called Best Lives. And this podcast interviews researchers to find out more about the story and the motivation behind their work. Another example is that we recently wrote an article on this research for the magazine Science Vega. And this is distributed to high school students, mainly in the UK, but it's online, so accessible to everyone, and aims to open up access to cutting-edge research for young people. And this article is a question and answer style piece with the team, and it explains each of our roles in this process. Next, we'll also be writing an article for the special access issue of Frontiers for Young Minds, where we'll translate this study from a scientific article into an article which is understandable by 8 to 15-year-olds. And this is with a team of scientists, clinicians, and people with lived experience. We also have plans to make proteomics and precision oncology more accessible to children and their families. And to do this, I'm excited to work with Karen Haas and Dawn Pickering, both people with lived experience and theme co-leads at ACCESS, to produce an accessible animation or booklet. And this will make use of some of these illustrations I've shown you at the start of this presentation as a starting point, which are narrated by an extra member of our team here that we call Pip the Prospector. And this may also link with the ACCESS Book Club, which is co-led by Karen and Trisha. So finally, those who know me or those who went to the ACCESS annual meeting will know that my favorite way of sharing and exchanging knowledge is using poetry. And in particular, using poetry as a way of forming a collective voice from diverse groups linked to cancer research. And as part of one of the learning sessions at the ACCESS annual meeting, which we had in March, I therefore invited the network to write down words in response to 3 prompts, which were, what does access mean to you? What do you want to tell the world about access? Why does Canada and the world need access? And how do you feel being part of the ACCESS network? And from this, I had over 80 responses, which I was very, very pleased with. And thank you to everyone for being so enthusiastic to participate. I read through these one by one and then used the words to inspire a collaborative poem to represent the collective voice of access. And I am now going to show a short spoken word animation of this poem, which is called Bridging the Future. Bridging the Future. A poem inspired by the words of ACCESS, a research network empowering the Canadian childhood cancer community to transform the health and experiences of children and their families. We are all threads, each one unique, from different worlds, speaking different words, hoping that the world will improve, hoping to be part of the reason, the how, to honour those who have had to fight, all seeking equity, breaking silos for children with cancer. ACCESS is the bridge to help us connect. Barriers abound, 2 mountains never meet, but 2 people always will. Je tiens un morceau de casse-tête, et toi aussi. Let's collaborate. Rivulets to river, a confluence flowing. A collective force, paddling together, upstream. Guiding, inspiring, empowering, discovering. Some have seen deeper seas, others have travelled farther. Creative minds intertwined, weaving pathways for the future. Pathways of hope and possibility, advancing care collaboratively, advocating for innovation to cultivate impact on what matters most. Can comes before cancer, et ensemble nous pouvons comprendre de tout. In a community of shared purpose, a rare space that feels like family away from mine. Together, we come from different worlds to imagine a better world. A coalition of wisdom, commitment, and experience, bringing change through synergy and opportunities where none were before. access to a better future for all children with cancer. So to finish, I would like to highlight the many people involved in the work we have shared today. And this is really only possible because we all work together in the Access network, from people with lived experience to clinicians and scientists from across Canada. And to quote the poem, we're bringing change through synergy to children with cancer. Thank you very much for listening. If you'd like to read any of the materials I mentioned, such as the podcast or the high school student magazine, you can find our lab website at the QR code here. And the rest of the Prospect team will now join us, and we're happy to answer any questions you have.
Thank you, Kirsty. That was wonderful.
Yes.
Thank you for everybody on here. We'll now go into our Q&A.
Yes, thank you, Kirsty, for this wonderfully outlined talk. And I especially liked the knowledge translation animation that you showed at the end. The Q&A function is now open, so feel free to share your questions and I'll direct them to Kirsty and the team. So I'll just give it a couple seconds to see some of the questions come in and we'll get started. Can see that people are writing, but it's slowly trickling in. Okay, so our first question is, so how will ultra rare cancer research and treatment benefit from proteomics?
So using proteomics, we can find if there's any vulnerabilities in particular rare cancers, in particular patients, but also across patient cohorts, that can be targeted by drugs which are already accessible to children or already have safety information in children. But in addition, as we analyze more patients, we may be able to find similarities between rare cancer types, which then can either lead to new drug development or approval of new treatments for specifically rare cancers. I don't know if anyone else in the team wants to add anything, please feel free to jump in.
Yeah, maybe I add one quote really from a cancer patient here at BC Children's Hospital several years ago, Michael Coccioni, who sadly isn't with us anymore. But he really, very early on as a teenager, saw that the cure to an individual kid's cancer can lie in the individual patient, in the individual kid. And I think especially for rare, very rare cancers, this is the key to unlocking a treatment opportunity for these children, is by looking very deeply into the functioning of their tumors. And that's what genomics and what proteomics allows us to do. Of course, in all of this, we have the economic balance, and we can't really weigh up the value of a life against any monetary one. But as a society, that's where we try to do both. We try to do all we can and the best we can for the children with a rare tumor where the standard answer is just not the right answer. And then from this, we try to learn that for the bigger groups, we can do it much more economically, that across the board, we can really give everybody access and enable hope for everybody.
Okay. So the next question says that you mentioned that those patient samples were luckily flash frozen so that you can test on it. Do you recall if they were flash frozen or viably frozen? And if flash frozen, how were the samples thawed before later testing?
So for the settle case, the samples will be viably frozen. So they need to be frozen uh, in a particular way to keep to mean that when we thaw the sample, that we can then the cancer cells then can be cultured or grown in a chicken egg in the lab. Um, sorry, what was the second part of the question?
So if it was flash frozen,
Yeah.
how were the samples thawed for later testing?
Um,
Or frozen thawed for later testing, sorry.
yeah. Yeah. So having I maybe should have mentioned that, but having samples viably frozen is very important for us to enable us to do the testing of the drugs in the lab, which we then find by proteomics. And this is something that we're working with James Lim, who I mentioned, and the pediatric preclinical modeling program to make this more widely known and to set up standard protocols. So freezing tissue in a viable format is routinely done because that really helps us validate what we find in the lab before it's presented to the hospitals.
Thank you.
And so to the question of flash frozen, we have done initial studies, or James Lim has done initial studies together with John Bush in if we can resurrect flash frozen tumors. And on some, this works to some degree. It needs more work, but the general understanding is that if we can get them well preserved in a viably frozen way, then we get a much more accurate information out, because in the flash freezing, we will likely pre-select cells for a particularly robust phenotype that is not maybe reflective of the whole. And I see John is coming on here. Please comment further.
Yeah, I'll just add, it's been exciting to try to find ways to make this viable freezing an acceptable pathway for a pathologist. We often are set in our ways. We don't want to increase the number of things we have to do when we are handling the tumors. And Dr. Jennifer Chan and I have worked to pilot a method so that no special techniques are needed, no special containers are needed for viably freezing tissue. And I use that royally. Dr. Chan has largely helped spread this protocol out to other hospitals. So going forward with the ACCESS group in our biobanking component within it, we are trying to encourage the availability of viable cryopreservation, or saving the cells so that they can be thawed alive at a later date, to sites that don't have specialized apparatuses like we luckily do here at Children's in BC. But everywhere patient access is able to potentially have viable cryopreservation.
Thank you. So the next one's not a question, but so a listener wanted to say bravo, Prospect team. They also wanted to applaud you for the way that you presented your topic to a lay audience and that you did it wonderfully.
Thank you.
Yeah. And the next question we have is, how necessary is it to have a control cohort to be confident that the increased expression of a specific protein in a patient's tumor versus their normal tissue is significant?
Yes, that's a it's a very good question. Um, so the ideal is that we will have a patient cohort for every case, but this requires having TMAs, so the tumor tissue microarrays, for lots of different pediatric cancer types. Perhaps, John, you want to jump in here because this is your area of expertise. I think we um, it helps us be increase our confidence that a protein is particularly high in the individual patient. Um, but of course, we may also find we might find protein targets which are common between cancer types. So yeah, there's some nuance there, but we try to include it when we have a TMA available.
Yeah,
Yeah.
I'll just add, it's it's great in today's healthcare systems where clinicians are trying to obtain tumor tissue for diagnosis in as little minimally invasive way as possible. So we're often getting just sort of needle biopsies, not receiving a portion of the tumor that we can then work with. So we often actually don't have normal tissue for that patient to compare it to. Who knows what that patient might be expressing or how their body might be that's different from another person. So we are trying two things. One, we're trying to using our archival tissues that we have to keep build these tissue microarrays so that we know what the normal tumors or a distribution in the tumors might look like. But also with Philip and Christy and Verena, we have a student who's building a normal tissue microarray, both in development, in sort of fetal development, because a lot of tumors go on to express proteins and genes that were actually present in fetal development, and your body shuts it down. And one of the reasons tumors grow is because it kind of reactivates those growth proteins in the tumor, as well as different age spectrums of normal skin, liver, just from other samples that we've had within the lab here. So we're going to have a very robust tumor TMA or tissue microarray, as well as normal tissues, and we can compare what that tumor might look like compared to a thyroid of a 5-year-old and a 12-year-old is something different happening there. So we're pretty excited about that option.
Thank you. The next question says, regarding leukemia or lymphoma, is it feasible to develop a test for the protein, either using serum or plasma? And is it fair to assume it depends on the availability of validated antibody?
I might hand that question to you, Philip.
Yeah, sure.
Yeah.
We can absolutely also do the same analysis on blood cells on leukemia or lymphoma cells extracted from blood, isolated from blood. We can also look in serum and plasma, both of leukemic patients, but also of solid tumor patients, where we might learn information, and that's something we're actively being diving into. The benefit of the methodology we use here, mass spectrometry, is that we don't need a validated antibody against a particular protein. We don't need to know the protein that is changed beforehand, because the technology allows an unbiased quantification of thousands of proteins at the same time. And so this is really a huge benefit in this. If one finds something that is consistently in a specific cohort of interest, one can develop it into the more established antibody-based tests. But for what we are doing here, we don't need this. So it can absolutely be done on leukemic patients as well. Yes.
Thank you. Our next question says, so we know that genetic sequencing can be expensive for each child, but we also know it could save more children and cause less side effects, and ultimately cost taxpayers less by reducing likelihood of relapse later. etc. So is proteomics even more expensive? Or how do we make the financial case for ROIs to the government still paying for all treatments?
Very good question. Very important question. And uh, I would say the financial case is pretty much the same. The cost is the same as sequencing, or even cheaper than, I shouldn't say even cheaper, but or cheaper than sequencing. Of course, by adding another methodology, another level of information that we are acquiring, we're essentially doubling the cost, or adding to the overall cost. I would say right now the biggest cost factor in this is that it's still in development. In the whole process around the annotation, the interpretation is still very much in development, and needs more formalization and streamlining. And that's where a lot of efficiency will come in, and which will drive down the cost to, I believe, a place where it becomes quite feasible to implement in our single payer system. And really then the financial case becomes the very same one, where the cost saved, especially in the pediatric cancer world, where there are so many years of life gained, and high quality life. And that's the other one that we are actively interested in, is how can we monitor for survivorship and ensure that we really get the best treatment with the lowest long-term impact on the patient. So I think that's where the healthcare system will benefit tremendously in it down the road.
All right. I think we have time for one more question. So it was great to hear the example you gave of an actionable result for a patient. So can you comment further from your own work and from the work of others in your space about how actionable proteomics data has the potential to guide treatment for patients?
So as I mentioned, we have analysed 5 cases so far across Canada, where we've reported the data to the molecular tumour boards. An area where we think this can immediately help patients is where, because having their DNA and their RNA analysed is standard practice in Canada for profile. We are looking to help patients where this analysis doesn't reveal any treatment options. So sharing the findings that we find from proteomics could reveal additional options. But for other cases, we also would like to ideally integrate the genomics and the proteomics analysis to provide additional complementary information to help clinicians guide treatments.
Yeah. Questions are still coming in, so I'll keep going. Is this limited to the age of 18 at this time? Like the use of?
No, it's not limited to, it's going, so it is right now embedded in the profile study. So it is governed by the approvals around this. And I think we're going up to young adults to 25, maybe even higher now.
A lot of thank yous and great talk comments being shared. There is a comment here that I would also love to share where it says they would like to say how proud of your team and to be a PWLE advisor on this project. Profile has helped save their son and when there were no obvious options left for them. And after a second relapse, so all childhood cancer patients should have options like this from the outset. And we also have one more question, and I think we can probably end off with that one. So MS-based proteomics has always been considered expensive, especially if you consider MS instrument costs, maintenance, etc. So while I do hope proteomics becomes a standard practice for all CAYA with cancer, do you envision this being sustainable?
Again, a very good question, and it is becoming sustainable. I absolutely see it with that way. There is tremendous development by the instrument manufacturers to increase the maintenance free time of the instruments and the throughput of the instruments, which together really leads to platforms that I believe get us into a good space in the future. So it will take a few years to really move this out of the research space into the routine clinical space, and we're working on this actively right now to establish an instrument in the clinical lab here at BC Children's Hospital to explore exactly these questions. What is the economics of this? What is the practicality in a routine environment? And we see these activities around the world becoming more and more common. So I believe the field is moving in that direction, and we'll see this in clinical labs in the years to come.
That's great. So that brings us to the end of our Access Presents session. Kirsty's talk will be available on the ACCESS website, like all previous ACCESS Presents talks. And so if you do have any further questions after today's session, you can find the emails that Kirsty has in her slide via the QR code and at the end of her presentation, or you can email the ACCESS Secretariat and we can get that directed to Kirsty and the team. So we'd like to, again, thank you all for attending today's session. And we just dropped a survey link into the chat as well for any sort of feedback you may have about the talk and the sessions as a whole. So thank you again.